you will very probably lose some of the maternal microbiota diversity. But if the baby of the mother that was born by C-section also gives birth in the next generation by C-section, then you can imagine that after 2 or 3 generations, you will definitely lose a lot of biodiversity. So the option of maintaining diversity would make a lot of sense because of all the functionalities that are protective and that you may actually lose if you reproduce that several times.
And one of the things you've spoken a lot about already in the podcast is biodiversity and diversity. How does diversity as a concept apply to the microbiomes of the body, perhaps most specifically the colon and a large bowel?
Yeah, so I was saying we are 50 trillion bacteria, many more microbes, in fact. And if you count viruses and phages, which are the viruses of bacteria, it's a lot more. And in terms of species, each individual's dominant gut microbiota is on average 300 species, which is really quite a big diversity. Only a few of those are actually conserved between individuals. We are tightly, closely, intimately connected with our microbiota, and it's really a part of us in that respect. And diversity matters. For example, when we compare the gut microbiota of patients in many chronic conditions with that of healthy individuals, we are able to identify distortion, alteration of the microbiota that we call dysbiosis. But one of the key features was a loss of richness or diversity. So much so that we tend to view this parameter, a rather simple parameter, as associated with higher risk of development of chronic conditions. Yeah, what more can I say about that? This is something that we would want to promote when we think of promoting a nice, healthy microbiota. And one of the ways to do it is to provide a diversity of carbon and energy source of the fiber content, for example, of the diet, if we only use the diet.
I really want to have a deep dive later on how to create the healthy microbiome, but maybe you could just unpack the concept of what is a healthy microbiome a little bit further. So you've talked about biodiversity. Is there a universally accepted definition that people can hang on to?
This is still debated at the moment, let's say. In many disease conditions, we've shown that if we compare patients and healthy individuals, we have a difference. And even though there is high inter-individual variability, we can still point at specific signatures in some diseases, species that are underrepresented, for example, that tend to leave the dominance of the gut microbiota. And we went all the way to document how these species sometimes were very important because they were conferring protection, specific protections. For example, the way they interacted with the immune system was very beneficial. And diversity matters, but the content of the microbiota really in detail will matter. And where are we today in terms of defining a healthy microbiota? So basically the idea we can have is that there is so much inter-individual variation. And I was saying there's just a few species that are highly conserved between individuals, that characterizing the healthy microbiota at the level of taxonomy of the composition of the microbiota will be a bit difficult. And so we've looked at that at the functional level. And when we look at functionalities, then we see much more homogeneity between individuals. And there are some functionalities that are really essential. I don't think that we've gone through everything that is important, but I can give a few examples. And one that I can give is, for example, the production of small molecules that are bioactive in many ways for the human body, that are important for our human cells. One of them is butyrate. So it's a short chain fatty acid that is produced by fermentation of dominant microbes in the gut. And some do it, others not. So you can have a microbiota that produces a high level of butyrate. And other microbiota will be deprived of the dominant microbes that do the job, and so they will not provide as much. And it acts as a source of carbon and energy for human cells. So it's very important for the maintenance of the integrity of the gut wall or the gut barrier, which is a monolayer of human cells that separates the content of the gut and the bloodstream, basically. So it's very essential that it's maintained as much in terms of integrity and impermeability.